Hello everyone and welcome to today's video notes. Today we're making a flip book on transcription. If you haven't already, you need to pick up this sheet of paper. You're also going to need your pencil, something to write with. Purple, orange, green, and then red. So pause the video now so you can go get your pencil, something that's purple, orange, green, and then red. Okay, now that you have your colors and your paper, first thing that we need to do is fold it in half. Make sure to crease the edge so it works like a book. Okay, so you should have something that looks like this. It opens up with this on the bottom. We're going to start on the cover of our flip book and we're going to have to label what we are learning. I'm just going to use blue because that's what I have closest to me. Okay, so first thing we need to label is that this is transcription. Transcription. This is going to be the first step of protein synthesis. First step of protein synthesis. And just want to note here, synthesis means to make something. So to make protein is what this means. We are also going to write down the molecule that we're starting with and the molecule that we're ending with. So we are going to start with DNA. And we are going to end with mRNA. And I'm going to add a cute little thing around that just so it stands out. And why not underline transcription? Okay, so we have our cover page for our flip book. We are then going to open up and only be working on the top right now. So another few things we need to write down before we get to the actual process of transcription. First, this is going to happen in the nucleus. So location equals the nucleus. We also are going to have a key. First thing we're adding to our key is DNA, because we're not going to be labeling this in our picture. Next thing we need to add is something that looks like an upside down comb. This upside down comb is going to be our mRNA molecule. Pause the video now, make sure that you have this down and your colors ready to go. Okay, now comes the fun part. We get to move on to our bottom notes section. Before we can start that, I'm so sorry the printer didn't make this look very nice. Please fix your 5 and 3 prime, because remember we are going to be working anti-parallel and everything matters here. We're going to follow the 5 prime strand down, up, down, up, down, up, down. So over here would be 3 prime. To make it anti-parallel, we have 5 prime on top. Okay, now comes the fun part. Going through and actually doing the notes. So, first thing that we need to talk about is this top thing. What is this? So we have a bracket around a segment of DNA. And from our notes in the past, we know that a section or segment of DNA is called a gene. So let's write down gene over in the top left. And let's just write the definition. A gene is a segment or a section of DNA. We're also going to write down our example. So in our example, our section of DNA is already opened up for us. Let's count how many bases long it is. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Our example is 12 bases long. So it's important to remember that in transcription, we are only going to be transcribing one gene, not the entire length of the DNA. One gene is going to code for one protein and one protein only. To start off transcription, you are going to need your green. I hope all of you know by now from sitting in a car or even learning to drive yourself, green means go. So if you see green at a stoplight, you are going to start driving. It's basically going to work the same way in transcription, but instead our green is going to look like a lollipop at the start of our gene. Let's label this little lollipop. Ooh. That little lollipop, that is called a promoter. And I'm going to underline that green. If you promote something, you start there, you want to keep maybe selling something if you're promoting a product, you just want to keep going. The promoter is going to be the cell's go signal. Transcription starts here. When transcription starts, we are only going to have one enzyme that's going to do all the work. And that enzyme is RNA polymerase. I know mine's a little hard to see. Do not draw anything right now. I just need you to watch the video. RNA polymerase is going to work very similar to DNA polymerase. They are both builders. So they will build the RNA strand, just like how DNA polymerase built DNA by adding bases. RNA polymerase kind of takes over the job of helicase too. So since this is just a flip book, I cannot draw every single step of transcription. We are only taking a screenshot here of the end portion. So I need you to use your imagination here for a second. I need you to imagine that this DNA looks like this. At the start of transcription, RNA polymerase is just going to be floating around in the cell and it's going to find a promoter. When it finds a promoter, RNA polymerase is going to attach itself to the DNA and start to unwind it, opening up the bases to be copied like so. Now, as RNA polymerase is opening up the DNA strand, it's also going to add the bases. So let's start at the promoter here. We're just going to unwind a little bit. And I'm going to pause. RNA polymerase sees that there are some open bases, and it's going to start to add. We have added bases. Again, you do not need to be drawing right now. In your vocabulary, you learned that A does not pair with T anymore. A is going to pair with a new base U, U being uracil. So let's start making our RNA. A pairs with U, C still pairs with G, G pairs with C, and we don't have any more space. RNA polymerase is going to continue to unwind and then fill in the blank spaces. So we have a few more bases available to us. We have T. Now T isn't going to affect anything on our RNA. T is still going to bond to A, or pair with A, sorry. A pairs with U, T pairs with A, T pairs with A, C pairs with G. We're out of bases, so RNA polymerase is going to keep going, keep going, keep going, exposing more nitrogen bases that we can match up. Made that one really tall. Some of these a little bit more tall. G is going to pair with C, C pairs with G, A pairs with U, A pairs with U. Now in this picture, I don't have it written down yet, but RNA polymerase is going to reach something that we call a terminator. Before we draw the terminator, first our backbone is going to connect our RNA, and I'm going to have you pause the video here and draw in our RNA. Okay, now that we have our RNA all drawn in nice and pretty, we're going to draw in our terminator, and we're going to need red because terminate means to stop. Again, I hope you know by driving, a stop sign is red. A red at a stop light means that you need to stop and not run it. At the end of our gene, inside there's going to be a red lollipop. And the red lollipop we'll label as the terminator. And I'll underline that red. Again, color coding everything that we're doing. The terminator is going to be the opposite of the promoter, so this will be our stop signal. And this shows the end of the gene. It's the stop signal. It shows the end of the gene. When RNA polymerase is at that stop signal, it is going to then leave and just go about and transcribe a new gene. Using your purple, I'm going to go ahead and draw in RNA polymerase. Instead of writing directly on it, I'm going to go ahead and label it up top. And write down the name of it. So it is RNA polymerase. And we'll underline this in the same purple that we just used. RNA polymerase. And let's write down its two jobs because it did do two things during our little demonstration here. The first thing that RNA polymerase had to do was unzip the DNA. By unzipping the DNA, it allowed RNA polymerase to have access to these free nucleotides down at the bottom. Without unzipping, we couldn't replicate. Second thing RNA polymerase does was to line up / build using the RNA bases. And what are the RNA bases? They are A, C, G, and U. Now we're almost done here with our picture. One thing to note here with the building, DNA polymerase we learned can only build in the 5' to 3' direction. Let's see if that's true for RNA polymerase. So we're going to start 5' here on this end. Go down, up, meaning this strand is 5', this strand is 3'. So let's say that's 3', and I'll write 5' up here. So if we know that this is 3', anti-parallel to that would be 5', and it builds to the 3' direction. So that still holds true. So we can even write here that it's 5' to 3'. Once this mRNA is completely built, again, RNA polymerase is going to leave. The mRNA is going to leave out into the cytoplasm through a nuclear pore, which is just a hole in the nucleus. And the DNA is going to wind itself back up, available to make more mRNAs down the road. With these bases and this shape, just want to make clear, and I'm just going to write it up top, mRNA is still made of nucleotides. The nucleotides that mRNA is made of is the same ones with DNA. It just has that one switch of a nitrogen base. So in case you need a reminder, here it is. Our 3 parts. We have our phosphate, our sugar. Our sugar changed though, because this is RNA, this is ribose. And then over here, this is our nitrogen base. So 3 parts of the mRNA nucleotide. This is the end of transcription. We're just making this molecule, and once this molecule is made, we can go ahead and move on to translation. Pause the video now if you need to take any of these notes. If you have any questions, please feel free to ask your teacher. We are here to help you. Thank you and have a good day.